Hello everyone, welcome back to my video tutorial for single-cell RNA sequencing data analysis. In my previous videos, I showed you how to use BP cells to save quantum matrix on disk for a large data set, and also I showed you how to use sketch-based large data analysis. Today, I'm going to show you how to use the reference dataset to map and annotate 1.5 million immune cells. I'm following the online tutorial. The reference dataset we are going to use for today's demonstration. It is the PBMC CITE-seq reference dataset. You can download it from Zeno's website. Here is the link. You can just click the download button to download it. I downloaded it already. So for the dataset we are going to use for today's analysis, we will download 3 datasets from the CZI Science website. The first one was produced by RN Etc. Then you can download the PBMC dataset. They sequence the PBMC from normal and COVID patients. And you can see here, this dataset has more than 800,000 cells. We can download this dataset from this link. And the second dataset was produced by Gene etc. You need to click this link to download the PBMC dataset. The third data was produced by Yoshida etc. You can see from here, they sequenced the cells from blood and also cells from airway tissue. We will use the first link to download the PBMCs, and you can see this dataset has 400,000 cells. So I downloaded all the data already. You can see here I named as RN PBMC, Gene PBMC, and Yoshida PBMC, and here the PBMC multimodal 2023 is the reference dataset. So now we can go to R, do the analysis. So first we need to load the BP cells package. We use the BP cells package to write the matrix data on disk. And we need the Seurat, Alzheimer's, and Tidyverse. So we set the options as Seurat V5, and also we set a maximum size for the future package. So the online tutorial, they analyze the 3 datasets in one go. For you to understand the process, I'm going to write the matrix data on the disk separately. So first for the RN dataset, we can load the data from my files directory as RN PBMC. Then we can create a folder RN PBMC to write the matrix on disk. If you have a look at my folder now, you can see we have a new folder here named as RN PBMC. Okay, so we saved the matrix data on disk. Then we can load the matrix data. We can have a look at the matrix data. You can see here in the dimensional names, the first one are the gene IDs. So we need to convert the ensemble gene IDs into gene symbols. Use the Alzheimer's function. Let's have a look at the dataset again. You can see now they are the gene symbols, not the gene IDs anymore. So now we can use the quantum matrix for the RN dataset to create a Seurat object. We name the Seurat object as RN PBMC. So you can see we have a large Seurat object here, RN_PBMC. If we have a look at the metadata for this Seurat object, you can see the row names are barcodes and the origin ident for each cell is the Seurat object. We have the N-count RNA and the N-features RNA. So in order to identify the origin of the cells from each data set, so we create a new column for the metadata named as the data set and label the cells as RN_PBMC. If we have a look at the metadata now, you can see we created a new column named as the data set and all the cells were labeled as RN_PBMC. So we created the first Seurat object from the RN data set. So next we will do the same for the gene data set. So we load the data in, then write the matrix data on disk. So now we can load the matrix data and convert the ensemble gene IDs to gene symbols. Then create a Seurat object for the gene dataset. We also add the dataset column in the metadata and label all the cells as gene PBMC. If we have a look at the metadata for the gene PBMC, you can see the cell barcode, origin ident, N-count RNA, N-features, and the dataset column. All the cells were labeled as gene PBMC. So now we can create the third Seurat object from the Yoshida dataset. We load in the data, then write the matrix data on disk. Okay, we saved the matrix count on the disk and then we can load it in the R. Now we convert the ensemble gene IDs to gene symbols. Now we can create a Seurat object as Yoshida PBMC. And also we add the dataset column in the metadata to label all the cells as Yoshida PBMC. So let's have a look at the metadata. You can see cell barcodes, origin ident, N-count, N-feature, and the dataset column. All the cells are labeled as Yoshida PBMC. So now we have three Seurat objects, RN_PBMC, gene PBMC, and Yoshida PBMC. We can merge the three objects into one, named as PBMC query Seurat object. Okay, you can see we created a Seurat object named as PBMC query. If we have a look at this Seurat object, you can see in total we have more than 26,000 genes and nearly 1.5 million cells in the merged dataset. And there are three layers presented in the dataset: counts 1, 2, and 3 for each dataset. So now we can save the PBMC query Seurat object. And we can have a look at my folder. You can see we saved the count matrix for each dataset on the disk, and also we merged the three Seurat objects as PBMC query. Now you can see we have the reference dataset and also we have the query dataset. This dataset contains nearly 1.5 million PBMCs. So now we can use the reference dataset to map and annotate the query dataset in R. So we don't need those data set anymore. Let's clear it. Then we can load the query data set in. You can see we saved the count matrix on disk. Even we have 1.5 million cells, the Seurat object only has the size 779 megabyte. So now we can normalize the data. So after data normalization, we can load in the reference data set. You can see in the data window we have the PBMC query data and also the reference data. Now we can use the reference data set to map and annotate the query data set. Let's have a look at the reference data set. You can see it has more than 20,000 genes, more than 161,000 cells. So if you see for the reduction analysis, you can see you have the APCA, AUMAP, PCA, SPCA, UMAP, and WNN UMAP. So we will use the SPCA reduction method from the reference dataset, and the normalization method will be SC transform. So we can find the transfer anchors between the reference and the query dataset. Let's find the transfer anchors. This will take some time to identify all the transfer anchors. Okay, we found the transfer anchors between reference and the query dataset. So we can use the transfer anchors to map the query dataset. And we will map the cell types level 1 and level 2 from the reference dataset to the query dataset, and the reduction model I just showed you before. We are going to use the WNN UMAP. So now we can map the query data now. Okay, so we finished the mapping and the annotation for our query dataset. So we can have a look at the metadata now for the query dataset. So you can see we have the cell barcode. Now we have the predict cell types and also cell types score, because we mapped to the level 1 and level 2 cell type from the reference dataset. So we have predict cell type level 1 score, predicted cell type level 1, level 2 score, and level 2. So now we can use the dim plot to visualize the cells. So the reduction method will be the reference UMAP, and we group the cells on level 1, and also we can split by the data set. Then we can see the cells from 3 different data sets. So let's run the dim plot. Because we have 1.5 million cells, it runs very slowly now. Okay, we can see the cells in the plot windows. Let's zoom in to see the cell clusters. So you can see we successfully mapped and annotated the cell clusters for 1.5 million immune cells. You can see the cell clusters from 3 different datasets. You can see individual cell clusters. We plotted it on cell type level 1. You can see monocyte, dendritic cells, B cells, NK cells, CD8 and CD4 positive T cells. Following the mapping and annotation, you can use the data to do more analysis, such as finding differentially expressed genes, identify signaling pathways between healthy control and the COVID patients. So I'm going to stop from here for today's demonstration. I hope my video tutorial can help your large data analysis. Please subscribe my channel if you haven't subscribed it, and also share my videos with your friends. Thank you, and I hope to see you in my next video tutorial.